Here we go. Now I think I'm recording. Okay, here we go. So this define here, right here is what tells the interpreter that we are creating a new function. So what is a function? So function has two main pieces. Okay. So one is the input to the function. Okay. So what goes into the function, and then what is the output from the function. So the output of the function is right here. So here, for example, I have this function called calculate BMI. And what is its input? Its input is what is in the parenthesis here. So the weight and the height. And the output is what is given by the statement return here. So the return is what gives you that output. So here, what we have is it takes in these two inputs, weight underscore kg and height underscore m, and then it calculates this quantity BMI, which is this very, very simple formula, and then it returns the BMI. Okay, so this is a very, very simple function, right? Because it's only one line of code that it's really helping with. But you can imagine that there can be more complicated functions. And for such, it's really important to break the code down into different pieces of block. And especially if it's a block that you constantly reuse, that is when you want to wrap it inside a function. Okay, does this make sense? So I want you to try running this piece of code here. So just this piece from here to here. Yeah, just put it in the Python cell and run it and see how. So here is the function, okay? And this line here, patient BMI equals calculate BMI 70, 1.75. This part is called calling the function, okay? So you're invoking the function. You're telling the computer to go find what that function is and evaluate it. Okay, so this is how we call the function. Was everyone able to run this code? Yeah. Okay, good. How much did you get was the BMI? 22. How much? 2.86. Is that what everyone got? Oh, okay, that's good. Now you have to input the numbers in the function. Versus it's too complicated to ask the calculator to ask you for the numbers. So this is a function. So you mean like similar to how we did blood groups yesterday? Yeah. Yeah. So you could totally do that. So you could have input the weight of the person, and then input the height of the person, and then it will ask the user for these two numbers, and then you can be like patient BMI equals calculate BMI, and whatever the input weight was, weight and height, you can put weight and height as variables now, and those will have the actual value stored in, and then you can print out the patient's BMI. And that command can be before or after the definition of the function. Yes. So the definition of function can be more or less anywhere, except before you call it, you better should have run it. Okay. So this is what you were suggesting, right? So let me just make a. So you were just suggesting weight equals input, enter weight in grams, and then you have height equals enter height in centimeters, patient underscore BMI is equal to calculate. So I'll copy this code now from here. So this code, for example, should work. So here, so it is suggesting the changes. Right. It's like you put like a variable there. Yeah. So you can put in the variable there. And here, the it is really important that, okay, so it's suggesting these because it's correcting the units. AI is too smart these days, but okay. So anyway, so here what's happening, right? Because I have assigned these two variables, weight and height, into those things, they now carry values. So now you can pass in those variables in. And that is also where the power comes from. Because now what can happen is you can imagine there are two functions. The first function's output goes into the input of the second function. And you have no idea a priori of knowing what that input will be, right? So it will probably get stored in a variable, and that variable will get passed in. Okay, so now I want you to try and play with this piece of code here. So this is a very simple call to get concentration. So just copy this code and try to get it to run. And then similarly, you can also create a function which converts Celsius to Fahrenheit. Also, you can create a function that checks if a string is a DNA string. So the system runs okay, or if when I'm in. No, it will run okay. Let's switch on the lights a little bit now. Oh wow, it switches on too many lights. Oh, Paul, this is in the previous day two exercise. Listen. Oh, I'm talking to the person online. But from Celsius heat. Yeah. Uh, I put in that string. Also, you put and parameters also matter. Like we have these it's the string. No, no, not that. It's not the input and it's states. Oh, okay. And then you might want to put an input like saying that enter temperature in Celsius or something like that. Thank you. Good. No, you can. Are you stuck on how to do the Celsius to Fahrenheit? How do you do it? It's someone. It's, oh, yeah, it's these two. Yeah, just so. You, you need to always do this. Oh, such a narrow bit of time. Yes. No, it's not just parentheses. So here you have, so if you remember this input always makes it a string. You have to make it an integer for you to do all these multiplications and additions. Oh, an integer? Yeah. So you have to do, if you just press and not that next line after that. Sorry. Yeah. You have, so you just type Celsius equals int of Celsius. Or float of cells. I see. So to define that. Yeah. So it's initially this data type called string. So string is just text. So it doesn't know how to do multiplication and things like that. I mean, there is, but it's still thankfully at least doing it. Okay. Yeah, that's it. Okay. So every time you have an input as a number, you have to specify. Yeah, if it's a number, then you have to tell it to the computer, okay, they will send in a number, which you will think is a string, but convert it back into an integer or a float or something. See, but if you word like text, I don't care. You don't need anything in code. Yeah. Yeah. So for example, so this same code, right? I could run it. And in Celsius, I could write my name. Yeah. Like this, and it will perfectly take it. But then it will say could not convert this string to a float column. But if I erase the float, then it's fine. Then it will stop. Then it will break down here. Yeah. Okay. Yeah. It will try to do something, but it will break down. Okay. Yeah. You're saying get Celsius temperature. So then you firstly you cannot have spaces in the definition function. Okay, so here this thing which you have here, that's the name of the function. So you cannot have spaces. So for example, let's say you call the function get Celsius. Yes. Okay. Now let's say you take in a temperature, okay, in Celsius. So now this is the input to your function. So here is where this formula goes. Temperature in one. So here, when you call it this, you know, so Fahrenheit equivalent is defined before. Can you copy paste this before written? Yeah, that depends. But actually, just remove it. Yeah, that one. Yeah, and the Fahrenheit. Yeah, it is. So again, in variable names, you can't have spaces like this. So I would put Fahrenheit underscore equivalent. Underscore underscore. So, this one. This one. Yeah, that one. Equals. So here initially you're taking a temperature, right? So this is temperature into 1.8 plus 32. So there's no Celsius anymore. Yeah. And again, underscore equivalent. Yeah. So now this is your function. Okay. So now you can call the function. So let's say temperature A. Yeah, equals get Celsius. This function get underscore Celsius. Yeah, just that is yeah, sure. Yeah. Remove that point five. Yeah, that has a call. One more delete. Yeah. In this temperature now put in some 25 or something. Yeah. Okay. Now print out temperature. Delete everything below and print temperature. Yeah, just print. Right, print. Yeah. Here. Okay, yeah, that's what's. This one you always have to put it down in the flower places. Now remove this line because there's no. Yeah, not right. Yes. Because the name of the function is C to F. Yes. So here it should not be C, it should be C to F. Sure. And then should not be again the name of the function, okay? So once you define a function, you cannot use that name for anything else. So that would use be Fahrenheit temperature or something like that. Okay. So can I use just F here? Yeah, you can just use it. But if I use change X, you cannot define it. No. So here you will not already put in the F. Because the input only needs to be Celsius. So you just return the F. Yeah, now it is. So what is this what is this? So it just means that you're printing the two decimals of the float. That's it. Yeah, so that's why you have I6 1 0. Yeah. I would prefer it if you define it like this. Heat function. The input is the temperature in Celsius. It's this continue and compare. So now you can print C to F of 0, for example, and that will give you something. Uh, doesn't need it. But see the formula is around should be plus 32. Uh, uh, so even define the function. But now you also have to call it. Well, only then will it print whether your thing is correct. But we have to be like print as is DNA of the sequence. Here, small p of function. So, and yeah, just yeah. You press tab it is offline. Oh, yeah, that's a good thing. Thank you. That's ASDNA. Oh, so you cannot name variable and the function in the same thing. So you are only allowed, especially this will cause many errors. So the function, so you have to call this say sequence or seq, something, some other name. Excellent question. So type of that. Yes. And there's a here. Yeah. So this one, it's block plug itself. So there you can use any names you want. I see. But nowhere. So for example, even in this block, you cannot call a new variable keys underscore here. Okay. If you ever do that, then it will just break down. You're using a substitute. Okay, wait. Give you pressure to convert to uppercase, and then you.
use yeah, to just correct for the law.
Nice, nice. Yeah.
That means that the red, I can erase it and leave the
Yeah,
green
great one.
one.
Great one. And so here you press accept. So you click
No,
accept.
you know,
So basically, so the green is what it's adding and red is what it's removing. So if you believe it is correct, you can press accept.
Can I ask for the synchrony, please put in
Yes.
um like how to make the pattern.
Uh, so either you can use the input function that we had last time, where we had like an input. Do you remember when we in the blood groups? So either you could use that, or you can just give it like you can just initialize it what you want. Like you can be like sequence equals string something. So how would you deal with it if someone gave you some small letters and capital letters?
okay, just do.
Yeah. Um, is everyone getting a hang of functions? So the idea is you have a function name. So again, this is one of the most important things, and these will cause you errors, which will be very painful to deal with. Never call a function and a variable by the same name. Okay? Always use different names. And one of the ideas when it comes to functions is, for example, if I were to define something like get Celsius, okay? So I'll just show you here. So if I'm defining a function called get Celsius, what I would usually do is do something like this. So I will not use any underscores or anything like that. Okay. So instead, I will just put in capital whenever there is a new word for me. Okay. So this is one trick that I use for naming functions. Whereas if it was a variable, I would use underscores and keep everything lowercase. Okay. So these are some tricks essentially, like to, have a fixed kind of naming schema so that you never call a function and a variable by the same name. Okay, so that is all I wanted to cover with functions. So now, please open the day tree. And also, I hope you have downloaded that massive data set I sent you. So I want to tell you a little bit about what that data set is. So this data set I got from 10x's website. In if you know what 10x is, it's this company that does the single cell RNA sequencing, and it has a lot of software for that as well. So this basically has roughly 68,000 cells. That's why if you see the dataset name, it will have 68K in it. So it's basically saying it has 68,000 cells. And what I did with the data set, so of course, like the data doesn't come to you in a very, very nice format, right? But I don't want it to be painful for you all to get started with it. So what I have done is I have taken that data in its original format and converted it into a nice CSV file, which is what I shared with you all. So I want to be honest, like it takes some effort to convert the raw data into a nice ingestible format like a CSV file. So do not be fooled into thinking everything will flow smoothly whenever you start data analysis. Like you'll have to do a lot of like, nonsensical steps sometimes just to get like data in nice format. But for now, I have already given that to you. So that CSV file is what we are going to analyze. Okay. Uh, and is everyone familiar with how single cell RNA sequencing data looks like? So essentially, I'll just quickly give you how it looks like. Okay. So it looks something like this. So you have the rows, which are all the cells, okay? And then each column is a particular gene, okay? So here there are some names which I don't understand exactly, but maybe you do. And then you see a bunch of numbers, okay, like ones and zeros and twos and things. So these are the counts, basically. So here, for example, there is one count of that the RNA related to that gene in cell number two. That's what this means. Now, I told you this has 68,000 cells. So this CSV, if you ever open it, it will have 68,000 rows. Okay, so it's a huge thing. And it has how many genes? So I removed a lot of the genes. So the original data had many more genes, but I filtered a little bit to keep the size a little manageable. So now it's only 700 MB or so, and I only left 5,325 slightly meaningful genes. Most other genes were basically zero all the time. Okay, so this is how the data looks like. Okay, does this make sense? Okay, so this is what you're going to try to work out. So what I have done here for you is basically breaking down into multiple steps. Okay. So the first step is, of course, setting up all the libraries to import. Okay. So this is really where the power of Python comes in. Okay, because Python allows me to use libraries that other people have written, use code which other people have written and freely published on the internet. And I can just import their code and use it as if I have written it, more or less. Okay. And this is where Python is really, really powerful because Python is so easy to write and there are so many people contributing to it that new software is written constantly every day. And especially when you are on things like the cutting edge, like this next generation sequencing and so on, like you will constantly be having new data and new tools to analyze that new data. And this is all most likely going to be written in Python first. So here, Pandas is one such library. So this is a very powerful library to analyze these kinds of like CSV-like files, okay? And NumPy is another version. It's a library where it allows you to perform numerical calculations with Python, but in a much faster way. And this import matplotlib. So this is another very big software which is written for plotting graphs and visualizations. And similarly, Seaborn is another visualization software. Okay, so this is the first step. Now, what I have for you is essentially comments like this. So I want you to use these comments to slowly fill in the code in each of those cells. Okay, so I don't have the code for you there right now. So here, I give you a lot of hints though. So here, df, if you want to read a file, it has this kind of a format. So all you have to do is go in and change the file name here to the actual file name. And so you might have already downloaded data into a computer, but you also have to push it to the Colab or wherever you're working. So you have to upload it there as well. Has everyone managed to download and upload to Colab? Yes. Oh, you're running it locally now? Am I supposed to be opening something else? So where were you opening it before? When I got the file, just opened in this window. That's a thing. It didn't open before. Yeah, so it never opens directly in Colab. Oh, so I can put this in there. Yeah, you put this in there.
I am trying to upload, but then it's not sure. But
It's actually.
it's just not sure.
Yeah. Sorry, what are you trying to put in close?
And the little cat.
Yeah. Oh, not here. So you have the data soon, and so you go in here. Yeah.
In my picture of this, I remember it is in the
I'm good in general, but if you can tell me what the local language means, please. So here it is one. Okay. Alright. Is there a
yeah. It's kind of and
but hopefully you know enough to um
I uploaded the file last night here, but it's not showing up.
yes, so if you upload a file yesterday now, it goes away. So it can, that's why you have to upload it again. So it only stays for the session, okay? So the data set you can keep on your local computer for however long you want, but on Colab, like you can only use it for that one session.
Oh, sorry, I thought I did it last night.
No, no, don't worry. Yeah. That's also fine.
it's cute.
Oh no, again, you're having the same problem. So you don't open it as a new notebook.
Oh.
So you already have this, right? So you go here. Now here you are.
Oh,
Yeah, so now here find the lines.
So there I call it basically combine the file thing.
Yeah.
But that's what that was called.
So there is a .csv that's
Oh, I see. Results.
so the file name has to be the full file name, even with the extension.
So once I download it, yeah.
Yes. Oh, it's still uploading.
Sorry,
Yeah, no,
I saw it last night.
no, no, good.
you know the brand name. I have a CSV.
Yeah,
We still have a sub app.
it's the same is not defined because you didn't run the set.
So I have to run all of this process.
Yes. Yeah, here the Wi-Fi is notorious. That's why I was telling everyone to upload it.
I didn't do it because I saw it.
Yeah, I should have said this before. This is my fault. Oh, this is- can wait for some people to upload. And once they upload, you can also upload this thing.
So in general, then if I'm doing any random work
Yeah.
in collab, if I exit, that all I have uploaded is going to be gone every time I
No, not everything is gone. So your whole notebook
Uh-huh.
with your code and its output will be saved.
Uh-huh.
What is gone is all the files you uploaded for that session. So if you uploaded data into Colab for that session, that is gone.
So if I'm doing work on collab, then every time I have to upload
The files,
the
yeah.
file.
This is the main disadvantage of Colab.
I see.
But usually, like because it's an introduction course, like as you saw, we didn't upload too much data. It's the first time we're uploading.
So is there a server that you can go and keep your data
Yes.
accessible?
So the usual way you do it is you have the data available on some server, and from Python you can download the data anytime you need it. So you'll just open, have a few lines saying download all this data
Okay.
as the first line in your notebook, and then it will download all the data.
And that server can be anything. Can be a hard drive.
And yeah,
Doesn't have to be a specialized server.
no, it just can be anything. It just needs to be hosted.
Okay.
How many of you were able to upload the data into the Google Colab? You were able to upload, but you were continuing to upload. Yeah, I think slowly all of you will manage to upload. So, to be honest, this is also part of something that is a realistic view of how data analysis goes, right? Because often what you're waiting for with large data sets is downloading it, uploading it, processing it, waiting for the input to output to come and things like this. So
People generate their data sets any way they want. So what you were saying is part of the job is when you open the data to understand how they organize the data.
exactly. So
Like
yeah, so when this original data was, I got the version, right? So it was written in all sorts of weird ways. Like there were some random things, like for example, every cell has its tags, and then like the genes have different names and so on. So then I had to match all these things. Like I removed all the cell tags because I don't really care so much about what the tag of the cell was.
Oh.
I only care about that matrix. And so you have to clean and you have to figure out what is it that you are interested in, what is it you don't care about, and you remove all that. And then, for example, there were many more genes. There were 20,000 genes. I removed them to just 5,000 genes, which I basically was trying to like brute force compress the data set so that it's easier because otherwise it was like 3 GB or so. And that would have been even more painful for you all to deal with because then you'll have to break it into 4 files and then process each file separately and things like that. Like you can already see how painful it is for 770 MB.
Yeah.
So you just imagine the whole data set.
So yeah, you know, since I've
Sorry?
since that you have it,
Yeah.
uploaded, and if you could, you hit run this just through, is it?
Yeah.
Let's will show you something,
It should just read it.
because I could just use the.
No, so it takes some time to read it, okay?
Okay.
So that's all part of the thing. So if I gave you a 10 MB file, it would read instantaneously. But because it's such a huge file, it will take time to read. That's perfectly.
This is the card, so is it?
Yeah, it's uploading. You see that small.
Oh, yes, I did not.
Yeah.
What do you want? You can use them.
Um,
Oh, you're almost uploaded.
Oh.
See, it's uploading here.
And the time
I remember last year, yesterday, the entire.
Yeah.
Of this one, who made it up?
So you can try to do it, but it will probably not work because it's a huge file. So you can't just open huge files like that. So what you did last time, but you just clicked on this, double click on it.
Yeah.
I don't know if you did it. See, it says it cannot cut. So you have to find other ways. And one of the ways is to use this function called head.
Yeah.
So what you do is you put df
Um,
dot head and it will show you the first few rows. Like,
just below.
yeah, it shows the first 5 rows.
Okay, he doesn't know.
Ah, nice, nice. What? No, it's here.
Oh,
So it's uploaded. No, you don't open it anymore. It's because this is a large file, you can't open it like we opened yesterday. See, here it says file is too large to display. So you have to use Python to read it and open it and things like that. Now can you try uploading? I tried this. Yeah, can you just download? Yeah. It's 0 bytes, that's the problem. You did not download that. Okay, I think most of you have managed to somehow upload it. So the first part of the code was just this. Df equals pd dot read CSV of this. So this pd is what? Is the pandas. Exactly. And this is a function that someone has written, read CSV. So what it does is it takes in a CSV and reads it into a nice table-like structure, which people call a data frame. Okay? And that's what you're seeing here. So these are the row indices and these are the columns. Okay? And the way you so yesterday, you just clicked on the CSV file and you saw a nice, beautiful image of the CSV file. Now you're not going to be able to see that because it's a large file. So you need to specifically query and try to see how it looks like. So one of the ways to do that is this function called df.head. So what it does, it will give you the first 5 rows of your data. Okay, and it will tell you how many rows and how many columns there are. Okay. So here, for instance, you see there are all these genes, and then there is this weird column called unnamed zero. So this is actually my fault. So I made a mistake in processing because of which I introduced this new column. Okay. So now what we are going to do is we are going to drop that column. And this is how you drop columns. So this is a very important step in data processing because, as you can imagine, not all genes are important to everyone. Okay, so most people are interested in certain specific genes or certain specific pathways and so on. So what you want to do is ideally drop everything else. Okay, so here I'm just dropping this unnamed zero. So it calls the column unnamed zero. So I'm saying df.drop columns equals unnamed.zero. So this will just drop that column. And then if you again do df.head, you should see that that column is gone.
Yeah. F equal to.
Mhm.
With then
So here what I did was, if you see this slide, in the before this data frame had this weird column here. Now what happened is I dropped this weird column. So now this is a new data. the same data frame with this column row. So now if you look at the head, so df.head also allows you to take 10 rows. And so you see that unnamed 0 is gone. So now it's only these.
So, so, we could assume of the function you for some kind of train in the data to release the VN. If you can do, and from here, just for we will just see the
Yes. So when you do some operations on the data, so then you have to do df equals modification of the data frame, okay? So that your modifications are stored. Okay, because if you just modify the data frame, then it is stored temporarily but forgotten later. So that's why you have df equals you have to drop one, because then that modification that you have made is now stored into the data frame. Right?
I think I wrote the fix.
So remember, if you have already run this code once, what will happen is your df has now changed and it doesn't have this column unnamed 0 anymore.
Yeah, and that's what I got this error.
Yeah, so then you can't keep dropping, right? So let's go ahead here. Let me just run this. See, you have already dropped it. So you can't double drop it.
Oh, that's the error.
Yeah, that becomes an error now because now there is no such column because you have already dropped it.
So basically, I can just run this here and then go back and check again.
Yeah, essentially. Or you can just put this code again in the next cell and check it.
I see.
Yeah, so this is actually a very good point. Okay, so some cells, until now, every cell you ran, you could run it like any number of times you wanted. Okay, but now, for example, here, if you try to drop these columns multiple times, right, it's going to error because the first time it will see that there is this unnamed 0 column and I will try to drop it and it will drop it. But now, if I try to run this again, what will happen is it will not see this unnamed 0 anymore because I've already dropped it. So if I try to run it again, it's just going to throw me an error saying there is no such column. So this is another way of printing some rows, okay? And I want you to know this because this is quite useful. Instead of df.head, you can do df dot ilog, okay? And then you put in whichever row to whichever row you want. So here this is saying 0 to 10, 10 excluded. So this will print row 0, row 1, row 2, row 3, row 4, row 5, row 6, row 7, row 8, row 9. Okay. Now you could also do def.ilog. Let's say you're interested in the cell number 100 to 120. So you can be like 100 to 121. And that will print the gene expression data of cells 100 to 120.
So the head is just consecutive. You the first 10.
It's gives the first
But iLog allows you to do the range.
exactly. It allows you to put in a range. And this is really, really helpful because as you saw, you can't just open the CSV file anymore. So you need to have a way of just getting that little peek inside the CSV file to know what the data looks like. Okay. And by now, you probably also saw that there are a lot of zeros in here. Okay. And this is where it becomes very important what file formats you use and everything. So here, a CSV file format is a horrible file format to save all this data because it's mostly just filled with zeros everywhere. Okay. Instead, I could just remember to save just these numbers and where they exist, and that's much more efficient. And that is usually how the format is actually saved. But to process it, sometimes you have to get into these kinds of bad formats.
That's a colon, that's not a comma, right?
Which one?
0 to 10, 0:10.
Yeah, this is 0:10. Yes. Okay, and let's take a break here. Oh, by the way, whoever is taking this for credit, you will need me to, I will need you to run all these code and then export it as a PDF and send it via email to me. So now I will pause the recording. Oh, I think I'm able to hear everyone all of a sudden.
Hi.
Oh, yeah, I can hear you. Paul, nice. Okay, that's great. Okay, I hope you all have been able to uh, upload the code, to upload the data to Colab and so on. So let's keep going. So I'm sharing everything and I'm recording. Okay. So I now want to show you a really, really useful function, which is a must if you're ever processing a CSV file. It's this thing called df.describe. Okay. So what it does is it's really wonderful. So it will take each and every column and it will give you some statistics on that column. Okay. So for instance, try running this code and you will basically see a bunch of numbers. For example, it will tell you the count of the column, which is how many entries are in the column. This should be 68579. Those are the number of cells we have. And here it also tells you the mean. So what is the mean of mean expression of that gene across all cells? And then there's the standard deviation. There is the minimum value. There's the 25th percentile, 50th percentile, 75th percentile, and the maximum value. Okay. So it's a very, very good tool to get a rough idea of the distribution of the gene counts. Okay. So for example, let's look at this NOC2L, right? So what do you see here? Most genes, most cells have no gene. This is basically not expressed in most cells, okay? Because you see, even the 75th percentile is just zero, okay? But if you see the maximum, it's nine. What this means is there are many outliers, okay, on the right end of the expression. So there are many cells that are expressing quite a bit of this, whereas most cells are just showing nothing for this gene. Okay, so it shows some of the interesting biology as well. So here again, it's the same with many genes. If you see, most genes have this heavy outliers. So these are these cells that are just going into overdrive and just producing RNA, and many other cells are just sitting there chill, being like, "Oh, okay, you're doing that. I don't care. I'm not gonna produce any RNA or whatever."
Um, it's that the parameters fixed, or you can, for example, put standard error or maybe
Yeah, so unfortunately for this function, these are all parameters fixed. But if you are interested in certain specific statistical things, you can also perform those. And I'm going to slowly get to that, okay? So now what I want you to do is think about some questions you can answer based on this data you have, okay? So I want to give you some basic questions that I could come up with, but I'm not a biologist. I was trained as a mathematician. So I'm going to be asking more statistical questions and things like that. So I want to show you some of the questions I came up with. And if you find this interesting, you can keep exploring it. But if there are other questions you want to ask, which are meaningful biologically to you, we can also figure out how to answer those questions. So, for instance, one thing that I always check with like this single cell RNA sequencing data is basically the total counts for each cells, because that kind of gives me an idea of our cells. There will be some cells which are really dead, right? Because they'll basically not produce anything. So one thing that I always do is I basically go and check how much RNA is each single cell producing across all genes, not just a particular gene. So, for example, you can produce graphs like this. So here what I'm plotting is essentially the total count for each cell and how many cells have a certain count. So here you can see most cells, um, like here, which is 7,000, have counts roughly around 1,250 uh, RNAs. But there are, of course, some cells which are significantly lower counts. Like here, there are things that are very close to zero. So they are essentially cells not doing anything. And then there are cells which are like on overdrive. So, for example, there are some cells which are close to 10,000 counts. So they are just producing a ton of RNA. Okay. So you can also make graphs like this. So I have small pieces of code for you to fill out. So here the main idea is what I want to do is I want to count the total gene count along a row. Okay. So how do you do that? So that's this function right here. Dot sum axis equals 1. So this function. Okay, so this is what allows you to take row sums. So this axis equals 1 means it's a row sum. Axis equals 0 means it's a column sum. But before we keep going in the directions that I have pre-guessed, are there any biological questions that you have that you think this data can answer for you?
Classic one is how many cell types, you know,
How many cell types?
how many, I know you take the top 100 genes, how many different groups.
Mhm. Yeah, so that's the clustering problem. And unfortunately, I have also thought of that problem, and this is in the last step.
Yeah.
So this is still already in the program.
So the question is to decide which one is the outliers here
How do you decide which one is the
in
outlier?
this graph. Okay, if you have a count with 10,000 genes while everyone has 1,200,
Mhm.
that's an outlier, but how do you decide the cut? It's
Yeah, but this is a more subjective question, right? Like, how do you decide the cut? But if you have some way of deciding the cut, I can help you filter this to remove those
so
outliers.
first, yeah, do that. So let's say if it has, I don't know, one standard deviation from the mean, it's an outlier.
Mm. But you see, like, if you see the distribution, these have this kind of like a power law distribution.
Uh-huh.
And when you have power law distributions, like all the natural things that you think from like normal distributions, right? All of them go out the window because what will happen is the median is so close to zero and the mean is just being determined by the outliers. Okay, so essentially, like near the mean, there is no cell that is actually producing any counts near that. So most cells are not over there. They're near the median or near those outliers. So you have to be a bit more careful with power loss for that reason. In that case, if you don't have any strong biological questions, I suggest following some of the questions that I was curious about. And then maybe that will give you some ideas, and you'll be like, hey, this is not the right question. This is the correct question, or some modification of that question should be the correct question. So you should see multiple three dots, like the three dots is the place where you fill in the code. Oh, sorry, I also forgot. Um, before we all do that, could you also fill in the QR code? So this is the feedback for this workshop. I think this is. Oh, I forgot and put it in a different file. Never mind. Was everyone able to access the QR code? Yeah, it should not be too long. I think I was promised that it shouldn't take you more than 2 minutes. So hopefully that is true. There is no data here, just previous the data. Yes. Yeah, just that. It will take some time, but it will come.
We run the delete card.
Hmm?
We delete the card.
Yeah, so I will get to know.
So basically, I'm assuming the for this particular set, there is a protocol people follow, see the count, see the div, route direct, all that. So we have
Yes.
something.
Yes. So I want to put in this disclaimer. So the kind of pipeline I will show you is not the traditional pipeline people follow, okay? Like what my goal is to show you real data and how to play around with it. Often what happens like that when you're in a lab and you get your single cell data, you already kind of know what is the end goal that you want. You're not going to go in and be like, oh, I have all this new data. What does it say or anything like that? You're just specifically interested in a certain pathway or specifically interested in clustering or something like that. And then there will be a very specific set of sequence that you will follow. And this you can take in one of these classes where there is these single cell RNA sequencing pipelines classes. So you could take that. But what I want to show you here is how to actually play around with the data and get your hands dirty and learn some biology in the process potentially. So here, this is, I think, hopefully what you see. So here it will say total counts equals three dots. So this is the part where you need to fill in. And then I have a lot of boilerplate code for you here. So for example, like what I have is the average counts. So that's just the total counts dot mean. So these are all nice functions that again come with the pandas library. So this total counts is now a data frame of average values, and you're taking its mean, you're taking its median, you're taking its minimum, you're taking its maximum. So for example, this is the kind of thing you could do if you are interested in some very specific statistics. Okay. So this is what it will print out. And this step here is like some visualization. So what this will visualize is roughly what are the total counts and what is the frequency of the total counts. Okay. And so the most important part is, of course, to fill in this code here. So how do you compute the total counts? And this is where we will use this function predefined, which is called dot sum along an axis. So it's very, very simple. So total counts is a new data frame, which is df.sum axis equals 1. So what I want you to do is print out how total counts looks like, okay? Using either df total counts dot head or something like that, and print out how it looks like, and then also check these out. What is
When you put access one, what is the access that it's analyzed?
exactly. So PF, right? It has two directions. Okay. One is the rows, one is the columns. So when you say dot sum, you have to specifically tell it, should I be summing along the row? Should I be summing along the columns? So here, what we are trying to do is we are summing along each row. Okay, so that's what axis equals 1 tells you. Sum along rows. And axis equals 0 will tell you to sum along the columns.
When I finish doing the sum of the row, now I sum. What comes into the same name, but only the different one, these two is different.
Preferably different name because then it will cause you problems. Was everyone able to print the total counts? You should see like a nice sequence of rows, and for each row you should see a big number. That will be the total count.
The graph. You wanted the graph pointer?
Not the gap, just print out total counts.
Okay.
Like just do print total counts or something. So in all this code, what I want you to always do when you don't understand certain pieces, just start putting print statements and see what is in that data structure at that point. Yes. So, okay. And once you have all those nice total counts, like in that nice format, this code you can always use, okay? So this code you can always use to create this nice histogram, for example. You just need to replace total counts by some other number, by some other quantity, and you will get that beautiful bins and the nice curve that fits the bins. And this is one of the most beautiful single cell RNA sequencing data sets there is. Most data sets are more faint. They don't look as beautiful as this. Like here, you can very clearly see a nice power law kind of distribution for the total counts. And that is still true in other data sets, but it's so nice. Like I'm very surprised how beautiful this looks.
So when you say histogram, you will know what to do basically.
Yeah. So if you ever need to plot histograms, all you need to do is use this part of the code. So this code will just plot your histogram. You just need to figure out what its total counts will be. Like in your case, it will be something else. If you replace that by the quantity of interest to you, that will plot the histogram of that quantity. Yeah, so part of the reason I'm showing you all these graphs, even though I'm not telling you how these are generated and everything, is because I want you to see like how what Python can do for you as well. Like it can create all these nice graphs, which are more or less like ready, like almost publication ready. Was everyone able to get this working? Any questions?
Are you able to export that as like this separate file?
Yes, yes. Basically, if right here, again, I am a strong supporter for use of AI. So if you just ask AI how to save this graph, it will just tell you. So the code is very simple actually. It just goes plt.savebig and then the name of the file you want to give it, and it will save that file into your computer. In your case, into your Colab folder. And there are options to save it into PNG, JPEG, and SVG, like depending on what formats you want and so on.
And for the color we have, yeah, the color we have.
Exactly.
Color pixel.
Yeah, so here I chose red here, but you don't have to be red. And similarly here I chose this weird color. I mean, I didn't choose this. This is AI choosing it, but
The program allows you to pick from the palette. Okay.
yeah, exactly. So you can choose any color you want. You can also play around with these options. If you remove the KD equals false, you will see that this curve that's fitting the histogram from the out, it just goes away.
And almost.
And then you can remove this median line that I put here. Yeah, so there are lots of options, and this is not even like scratching the surface. Like you can change the title, you can change the X lane, Y lane, and you can add like many more things. This is like a barely um like this is like one of the most minimal graph examples. You can make much more complicated graphs.
Like the new map will come from this.
Yes. So I wasn't able to generate UMAP because I did not have enough computational resources, but hopefully you will get to generate UMAP. So that's the last step. And then one of the important things that you need to know is when you're dealing with large amounts of data, you don't want all the data that you have been accumulating to stay in your RAM. Okay, you want to periodically clean stuff. So one of the ways that you do that is via this code. So this is basically a garbage collector, and you just keep doing gc.collect. So maybe it's not that relevant for you because you're using Colab and it has a lot of resources. But for example, on my computer, I constantly have to do this because it's a huge file still, and it takes up a lot of the RAM, and like my computer just crashes if I don't keep collecting garbage. So another thing that you can look at is basically look at um, how many genes are expressed, okay, for each cell. Like in this case, for example, you saw that most of the times the genes are just zero, they are not expressed, okay? So one natural question is if I take a cell, how many genes are expressed on an apple? Okay, so that is what this exercise is trying to do. So I want you to go over it a little bit, think about it, and ask AI on how to do it. And then I'll tell you how to do it. Yeah, this is a lot. Oh, yeah, that's why. Have a name zero still. I don't think it had this code, but maybe it's different. Yeah, so this one will be it's the names of things in the dictionary. No concept. We go check the actual syntax. I just got confused now. Oh, it is that. So now I want to show you another really interesting operation you can do on data frames. So if you see this line here, where I say genes detected equals df greater than 0 dot sum axis equals 1. So what I want you to do is I want you to first print out just this portion. What does df greater than 0 print? So this whole kind of process with using data frames is essentially what's called vectorization in modern terms. So what we are doing is instead of using for loops, so if you wanted to say count how many genes are expressed for a cell, what would you do, right? You would go and look at a certain row, and in that row, you will go over each column and check which column element is greater than 0 and which column element is less than 0. So that's what you will be checking, right? That's how you will count the number of genes that are expressed for that cell. But this is extremely inefficient, as I told you with Python. Like the for loop does many more things under the hood than it actually needs to. And this is where it's important to use already existing functions. So here when you say df greater than 0, if you try printing it, what you should see is a bunch of true or false. Okay? And basically it will tell you true when it's actually greater than 0 and false when it is less than 0. And then if you do dot sum axis equals 1, then you're going to sum up along the rows of all the truths. So the true is essentially truth value is treated as 1 and false is 0. So if you add up all the ones, that's going to be exactly the number of genes that are being detected for each cell. So definitely suggest printing out just df greater than 0 to see what it does. And then I hope you can see what this dot sum axis equals 1 will do from there. Do you all print df greater than 0?
Yeah, it is a basically column with two of.
Yeah, exactly. So you'll get a calculation.
It's just this,
I don't think you can just do that. Wait, I didn't say df is not defined.
so
I think df is not defined. Wait, what happened to the data?
just,
Did you have to reload or something?
no, I just try the get issue function. This one.
Oh, and it collected everything. How did this happen? It's not supposed to do that.
Yeah, I just try this one and go over.
Yeah, I took everything out now, so you have to read that from the beginning.
So the process count how many genes have a value greater than 0 for each cell.
So df greater than 0 will tell you whether that gene count is greater than 0, right? So that's true or false. Now this dot sum axis equals 1 is counting along each row how many truths are there.
Yeah,
So for each cell, it counts the number of genes that are greater than 0.
yeah, but some cells have a different number, has 4, 5,
Yeah.
6.
Yeah.
So it's not you have 469 genes. You have like 469 prints along that route.
You have 469 non-zero entries along.
Yeah, and in one cell you can have number 5.
Yes.
So that would be
Yeah. So it's not that you have 469
gene.
exact counts of RNA.
Yeah.
It's those number of genes are active. So like for example, one RNA count could be 5, one RNA count could be 100, and so on.
That would end up to that 100. Oh, I see. It transformed.
So
I see.
in the previous case, we actually counted the total RNA
yeah.
counts, but here we are just counting how many genes are showing.
Because what you said, false is 0, true is 1.
Exactly.
So you can have 10 reads in a cell, but that's a 1.
Exactly. But what you will see, at least for this data set, is there is not that much difference whether I do a total UMI count or whether I just count the number of genes that are positive. Because you'll see that the number of unique genes detected is very much proportional to the total UMI counts that we did last time. So it's this very nice, beautiful straight line. Like that, this is why I say this data set is just so beautiful because everything just works out very nicely. Like all the things are proportional. It's this like when was the last time you saw data look like this beautiful, nice linear line here? Like this doesn't happen usually. So that's why this data set is very nice. So I also want to quickly walk you through what this code is doing. So the genes detected is basically the number of genes that are non-zero per each cell. And it's printing the average number of genes detected per cell. So that's roughly 500. And the max genes detected in a single cell, sometimes it's all the way up to 2200. And here we are just plotting what is called a scatter plot. So here you have two things, X and Y, and you're just throwing all of them as X comma Y onto this plot, okay? So here the total counts is the X, the genes detected is the Y, the number of genes detected, and now we are just throwing that onto the plot without any care for the order or anything like that. We are not trying to fit a line through or anything, just dots.
So I have a question.
Yeah.
So in the print F average genes detected per cell, yada yada, genes detected dot mean. So I see the colon there dot 2F. So is this then a dictionary with that? And then I don't know what the dot 2F is.
Yeah, so this dot mean here is essentially like a predefined function which takes in a data frame and calculates the averages. So that's this portion. And then this dot to f usually means that these are going to be float values. So it's going to be decimal values. And we are only interested in the first two decimals. If you remove that, nothing bad will happen. You'll just see more decimals. Yeah, but yeah, I don't know how much you have seen like the real single cell RNA sequencing data, but please believe me that this is like very beautiful data. Like this is one of the best sequencing data sets you will ever see in your life. So so one nice thing about this data set is that the cell clusters. So this is supposed to have I think three clusters, and all those three clusters were orthogonally validated. So they were first separated out and then sent through this single cell RNA sequencing pipeline. So this is one of the few data sets for which we actually know the cell types, like the ground truth of the cell types, which is kind of amazing. Any questions? Yes.
But I realized already.
Yes.
Can just go through all the things.
Ah, yeah, beautiful. This is how it looks.
But yeah, complete opportunity.
Yeah, because so because you just put d selected equals df presence, now it's only true or false. You need the whole thing dot sum max sigma. Now, wanted you to print this just to see what df grid has you, right? But because we are interested in how many number of such genes are, like we want to also add up how many proofs are. Okay, so maybe we can move forward. Yeah, so be a little careful apparently when you use garbage collector on Colab. For some people, it's just crashing everything. So, but I am unfortunately have to do it on my computer because I don't have that much RAM. So another thing that you could do is you can also look at the most frequently expressed genes. Okay, so these are genes with like very high counts. So I think in your version, yeah, okay. So essentially these three dots have to be filled in. So how would you take the total sum of each gene across all cells?
That's zero.
Exactly, because now we are going to do column sums because we are trying to see how much of that gene is being expressed in total. So this would just be df dot sum axis equals 0. Instead of row sums, now you're going to do column sums. Okay, so that would be your gene counts. And for top 20 genes, I want you to use AI to figure out how to do that. And then, of course, I'll show you the answer so that we can ensure that you are also getting the right one.
We need to more. Okay.
Yes.
Okay, nice.
So one,
So I want to give one small tip. So AI will give you something, right? So let's say AI told you how to count the top 20 genes. Okay. So let's think about how we can make sure that the code is working or working as intended. Okay. Any ideas on how we can check whether that code is actually working correctly? Print, exactly. Just print the top 20 genes. Okay. And go see whether those counts are actually high or not. Yeah.
so yeah, I want to think that it. Well, in this one, I think I just presented. Just 10 so just put some in the middle.
Yeah.
In case I'd like to have the whole 20 to make. But
Again, I should just asking AI. Let's see what it says.
So yeah, print. I have to print that. I mean, I have to assume that this should match the graph.
Oh, wait, how did it give you directly all the 20 nicely?
Uh,
Oh, but how it doesn't show all 20, it puts dots in between.
oh,
So one trick that I do is I print the 10
yeah,
and then print the next 10. So that's some tricks. So okay, it looks like AI is helping everyone. So I will show you the actual code. So again, so all the standard functions you can usually dream of, like sorting and everything, these are very fundamental programming operations. So they are already existing functions. So here you have gene counts, and now you're doing sort values. So this is how you start sorting. Okay. And now it's saying ascending equals false. So basically now it's going to sort it in descending order because ascending is the usual order. So you have to specifically tell it, don't sort it in ascending order. If you sort it in ascending order, it will be those genes which are the least expressed. That's going to be in the top. And then you want the first 20. So this dot head of 20 means you're going to pick out the first 20. And you should definitely print it out. And if it prints with three dots in between, one trick that I often use is I'll first print 5, then the next 5, and the next 5, and the next 5. That way it doesn't have the option to give three dots. And then the total UMI of the data set, that is the total RNA count for all cells and all genes, is just the sum of the gene counts.
so AI did something funny.
Mhm.
You can see df.sum.
Yes. So that is also perfectly fine. So if AI does df dot sum, so this is actually a very funny theorem in math, which is actually obvious, honestly. It's called Fubini's theorem, which tells you that if you want to sum all the entries on a matrix, you can first sum up all the columns and then sum all the rows. Or you can sum all the rows and then sum up those particular rows. Okay, so it is just one of these things. So here what we did, we first summed all the columns and then we are summing up the column sums.
Okay.
Okay, in df.sum, you are summing up potentially all the rows and then summing up those rows. So you'll still get the same answer.
I see.
So that's also perfectly fine. So this could have easily been df dot sum dot sum. And here again, but I want you to put in the axes just so that it's clear in your head. So there's a default one, which I forget whether it's the columns or the rows. But here, for example, you could do axis equals 1 and then dot sum.
What's the next 0? Oh,
Now, after that, there is no more axis. There's only one axis.
I see I see with the axis when you're telling it to do the rows after you did the column.
Yeah. And so if you run this, you should see something like this. Again, this graph is a classic code for plotting a bar plot. So here you just do something dot plot, and here it says which kind. It's a bar plot here. You can replace it with other options. Then you can see the total counts across all cells for these particular genes. So these are the top 20 most expressed genes. I have no idea actually what each of these genes mean. Do any of you recognize what these genes are?
RPS and RPLs are ribosomes usually.
Ah, okay, good to know. So RPS and RPL are ribosomal. Okay. What is this MALAT1? Do you know what this gene is? Like, why is it so highly expressed?
Uh, I,
These are supposed to be blood cells if it helps.
something called G cards. It can tell you easily.
Ah, okay.
One metastasis associated lung adenocarcinoma transcript 1. It's a cancer.
Some cancer gene. Okay, good to know.
It's highly abundant. Oh, it's non-coding RNA.
Is non-coding RNA. Oh, okay, interesting. So then why is it a gene? Anyways, this is not the time for my biology education.
Yeah.
Oh, drop the list in ChatGPT. Yes, and that's how you do it. Drop the list into ChatGPT, and it will tell you a list things about the genes and so on.
I mean, non-coding RNAs are coded by genes.
Ah, okay.
That's the DNA.
I see.
To be honest.
Okay, and here is one thing that I am a little bit suspicious about. This was generated mostly by AI. So it says there is this thing called mitochondrial. You can visualize cell health based on how much percentage of genes are mitochondrial genes. And apparently, mitochondrial genes start with MT. So I'm just so to me, this exercise is more like just an exercise. I don't know if any of the biology that AI came up with is true, but it's still fun in the sense that you get to see how to filter out only genes that have something in it. Okay, so like for in this case, they have to be starting with MT here. Okay, so again, let me see your version. Yeah, so you essentially have to fill in these two. So empty genes is the new part, and the counts is again similar to the code from last time. So again, ask ChatGPT on how to filter columns that start with name empty.
That is a thought. Of course, you have usable function on.
Oh, that's interesting to know. Even people you're using Colab, even though you're using fancy computers in the cloud, even it's crashing for you. Let's see if you are able to process this whole data set.
this first?
Yes, I will tell you again.
Okay.
So the first idea is you want to figure out all the genes that start with empty, right? So we are nicely going to store that in a list, okay? And if you remember from last time, whenever you see a for inside a list, that's basically what's called the list comprehension. Okay, so this could have been written in a more nicer way, which is you can be like for gene in df dot columns. So df dot columns is a list object that contains the names of all the columns of your data. Okay, so now what we are doing is we are going and looking at each column name in the data frame and checking if the gene starts with empty or not. And if it starts with empty, we remember it. Otherwise, we forget. So that's how we will have a list of all the genes that start with empty. And once you have a list of columns that you're interested in this case, once you have that, you can just do df empty genes. So here is a list of column names. And when you do this, you should try printing out empty underscore counts and also empty underscore genes. And what you will see is you'll only have genes that start with empty. And once you do this, empty underscore counts will be a data frame which will only have those columns that you're interested in. And this is how you specialize to very specific portions of the data.
I have a question.
Yes.
So it did exactly what you have there, except that instead of team, you just call for column. And
Yeah, it can.
so
Yeah.
column is a generic, so the computer knows what a column is, and it recognizes gene because your column starts
So-
a gene name.
No, so this name or something in call something, right? Here I can put in anything I want. So let me say this again.
I
So,
see.
so here, when I say gene here, it can be anything. I can put for psi in df dot columns. Okay, it's a name that I am giving, but this one now has to be related to this name.
I see. So basically, for an X,
For x in df dot columns, if x dot starts with empty, then remember the x.
I see.
Okay?
And you write genes so you can keep track of what you're doing.
Yeah. So definitely do a print here and also a print here where you do print. Actually, do this empty underscore counts in two steps. One first, just df of mount genes so that you can see that now you're restricting yourself to specific columns, and then do the sum so that you can see the empty underscore counts. And this is also perhaps a good time for a break. Okay, I am recording. There we go. Okay, so another thing that you can play around with, okay, is gene-gene correlations, okay? Because often, right, like the genes are participating together in certain pathways or things like that. So you can try to look at what are the genes that are highly correlated in their expression. So that's what we will be exploring here. And because if you try to look at all the genes, here we have roughly 5,000 genes, okay? And if you try to compute the correlation of 5,000 by 5,000, that's not going to be fun. So what I have done here is trying to do this only for like the first top 15 genes or so. But of course, you can, like if you have the enough compute power and so on, you can totally do it for the 5,000 by 5,000. I just didn't want to do it for this introductory class just because it will require actual computational power. So here, the first portion is essentially figuring out what are the genes that you're interested in. So here I'm just looking at the top 20 genes, and of those, only the first 15. I could have actually looked at all the 20. There's no reason to restrict myself to 15. And then this is a very useful function that I want to mention. So you take any data frame, and if you press .cor on it, like here, it will tell you all the possible correlations between every pair of columns. Okay, this is a very useful function, and it's just one line, and it's also heavily optimized. So if you try to do it on your own, like you will probably make a function which is much less optimized. And if you use this Seaborn, you can also nicely visualize it like this way. So one thing that's really interesting with all these gene expressions, if you see, all the correlations are quite positive, if you see. Okay, so you should also be able to generate this. All the correlations are positive. Like it's never like there is certain RNA being produced in massive quantities, so something else cannot be produced anymore or something like that. For some reason, these are always very positive correlations. Not, it's very rare to find a lot of negative correlations. Which is again very interesting if you think of it from a biological perspective, like why that's happening. I don't know good answers, but yeah.
Um, so you just pick the top 15 genes in the list,
Yes.
the top 20. So if we are interested in the, I see. So
So
we can organize the data differently and
.core and so for example, let's say you're interested in some very specific pathway, okay, which has genes. Let's say it's you're only interested in this RPL and RB genes, okay? So then you could go and tell your interesting genes to be genes that start with R,
okay.
like ribosomal genes. And you could look at that and then again do core matrix equals df of those interesting genes dot core. And then you will have the correlation for the ribosomal genes, if that is of biological interest to you. And if you're interested in, say, mitochondrial genes, then you could just look at those genes which start with MT, and then you will just get that. And this is usually a very good visualization to look at to get a rough idea of what's going on. And I just for fun, I also took many more genes, like 50 genes or so. I don't know if you can see the whole graph, but you can see, right? Like there is no blue. Like there is no blue. Like most things are positively correlated. Maybe they're not correlated, like you can get closer to zero. Like for example, things like this. Here you're getting very close to zero. Sorry, I keep forgetting. So there are these nice blocks, patterns where these are getting close to zero, but for the most part, they are positively correlated, which is very interesting. And these genes are all over the place, right? Or are they all ribosomal genes? Maybe they are all ribosomal, and maybe that's why. Yeah, anyways. But it's very interesting. The white portion corresponds to this FTL gene. I don't know why. And then there are these ACTB, HLAB. These are the portions which are the least correlated, but everything else is positively correlated. So it gives you all these nice, interesting questions as well. I don't know if it's really interesting, but it's interesting to me because I don't know any of the biology. So try filling out the code. I've already shown it to you, but you can make your own interesting genes and you can look at your own correlations. If you find some interesting correlations, please share it with me. I am always very curious.
If you want to correct, if you have one gene in mind
Yes.
and you want to correct it against the top 50,
Yes.
is that, and you can also write it in that.
You can do it in that way, but it turns out, unfortunately, that still, let's say you have one gene, you want to see how it correlates with 50 genes. The best way to do it still is you put those 50 genes and that one gene and create a data frame with 51 columns
You want.
and then just do dot core. So basically it will take pairwise everything, but it will also, as a part of it, it will also calculate the correlation of that one gene with respect to everything else, right? That will be one of the rows. And that turns out to be faster computing all pairwise correlations than using a for loop to compute just those 50.
So if I want to add a gene to the top 50 genes, I have to make another line of code to say, I want to add.
Exactly. And you can ask ChatGPT. And basically, now that you have said so nicely in English, ChatGPT will give you the correct way to add that particular gene. What is this gene that you're interested in?
My dad.
Okay, okay.
We all have favorite things.
Okay, okay, nice.
But I want to see if you can do both, have the top and then 30.
Yes.
I mean,
Yeah, Paul also points out I find it useful to spell out what you want in the comments. And this is something I also strongly suggest. If you write beautiful comments, the autocomplete will give you the correct code.
Where do you save your blocks of code? Where do you save your blocks of code? Functions that like if you're going to use something over and over, where do you save?
Oh, so if you're asking if you are building some functions that you will use again and again, so this you'll probably learn a little bit in advanced Python class where you can actually save things called Python scripts. So usually that's what you do. So you'll write Python scripts in your computer in a certain folder, and then you can access them from this notebook. You can be like, go find that folder, find me that function, and now apply that function. So that's usually what people do. But because we are doing elementary stuff, I didn't bother to do that.
Just interested.
Yeah. So if you're joining a lab or something, that's probably what you're going to do. You'll get some already written functions by someone, and then they will all be in script format, and then you'll access them from the notebook or something else.
I get it. It's not as simple, but it will like you said stepwise. So I said, yes, I think that you want to go stepwise for auto run. So I was going stepwise, and in the second step, it gets them.
Yeah.
But I get it.
Yeah, nice.
Thank you.
Okay, I hope you have all been able to calculate your own correlations that you like. If you ever get some value less than zero, I would be very interested. It's surprisingly very hard to get less than zero.
But then you can find genes that have a long coding RNA on the antisense strand.
Mhm.
Could be anticorrelate.
If you know any such genes, we can compute those two correlations and check.
The name, but that's how some regulations have.
Okay,
I don't know them by name.
yeah. Anyways, if you remember it later, just send me the name by email and I can check if they are negatively correlated. Sorry?
Are these blood cell data set?
Yes.
No.
Okay, so now in the remaining time I have, I wanted to show you some fancy stuff that people do with RNA sequencing data. So that's PCA. How many of you have heard of PCA before? Okay, many of you have heard of PCA. How many of you know what PCA is? Okay, people know PCA. That's great. That's great. That's wonderful. So PCA is basically about trying to somehow reduce the dimension of the data without losing much of the data. Okay. So for example, let's say certain genes, they're always zero, right? Then you can just ignore that column because they're not really doing much. They're just zeros. So that's the kind of rough idea. Okay. So we can do all this PCA and everything using Python. Okay. So I want to show you how that's roughly done. Okay. So here in this code, let me be here because I can. So the library that has all these machine learning tools that you will ever need is called sklearn. Okay. So if it doesn't directly run for you in Colab, you'll have to pip install sklearn. I can walk you through that a little later, but try running it, and I think it should work out directly. So sklearn is the library. And in this library, there are some sub-libraries called decomposition, pre-processing, and there are more. And from that, you can import the specific functions that you want. Okay. So here we are importing two functions. The first one is PCA. That's self-evident. And then there's this other thing called standard scalar. Any guess on what standard scalar is?
Uh, it reduce dimensions to a certain.
Not quite. So what is one of the most important things you need to do before you apply PCA? Normalize it. Okay, because if you don't normalize it, what can easily happen is just imagine, right? Like let's say you have a sphere, okay, but I just drag it out like this, like a big ellipse. So then PCA will just easily think that the other dimensions are just noise and it will just give you that elongated dimension. So you don't want that to happen. So often you normalize. So that's the first step here. So what we are doing is basically just normalizing that data into this reasonable format. And this you have to do before PCA. Otherwise, you are just going to like lose a lot of data. Okay, and then the next step is to apply PCA. As you can see, it's really, really easy to apply all this because it is written in such a nice format. So first you have to figure out how many components you want. Okay, so there are two ways. So either you say, I only want three components. Okay, that is you want to. So here, what is the original dimension, right? It's roughly 5,200 dimensional. So those are the number of genes. You can ask, oh, I only want 5 dimensions finally. I only want 2 dimensions or something like that. Okay. Or the other option is to given this information of how much variance you want explained in the data. Okay. So it's really funny, but it, this number of components can take two types of inputs. Okay. It can either be the total final dimensions that you want or the amount of variance you want explained in the data. Okay, so here that's why you see this 0.65. So here I'm asking, okay, come on, just explain 65% of the variance. Okay. But here, if you, for example, put number of components equals 3, you will get a three-dimensional matrix, not three-dimensional, 2D matrix with 3 columns. That's what you will get. And then the next function is dot fit transform. So if you just do dot fit transform of the scaled data, that is it. That's your PCA. And you have already performed PCA. Any questions for me so far?
Oh, I crash.
So now you will start hopefully seeing some crashes because you'll be dealing with a lot of data, so things will start crashing.
If it cracks, it cleans the garbage, so I have to use the clean garbage.
No, when it crashes, it cleans the garbage automatically. Okay, so maybe this will run, maybe this won't run. Let's see what will happen. So I'll keep going on what else we usually see with a PCA. So one is we want to always remember what is the dimension the PC has gone to. So in this case, the number of components is associated to 0.65, assigned to 0.65, which means now it's going to try to explain 65% of the variance in the data. So here you see that it requires 2,621 genes to explain just 65% of the data. So for some reason, all the genes are really important of the 5,000 that I gave you. And there's also a function called PCA.explained variance ratio. You can see there are lots of underscores here. So these are the kind of functions that are sitting inside, usually, like these are very deep inside the function. So you can again compute the total variance that it explains. So here it shows you that you have 2,621 components, and this explains 65.01% of the variance. And then you can also plot, and this is another plot people always do, which is the number of principal components on the x-axis and the explained variance on the y-axis. Okay, and usually what happens if you have very, very nice data, this is very nice data, I must say, but what can happen is basically what you will see is the variance will quickly go up to a very high value and then slowly increase from then on. So then people cut things off at that value of components because then they will be like, oh, the rest of this increase is just basically noise and everything else is meaningful. Now, again, like I'm not saying this is all statistically meaningful to do, but this is what is usually done in practice. So you'll get some graph like this. In this case, you can see that you do need a lot of principal components because all the genes are doing something meaningful to the data. So if you were if the Colab crashes when you're trying to run this, what I suggest is just initialize the data frame, okay? Drop the unnamed zero column and immediately run this cell, okay? So that way you get rid of all the other stuff happening in between. And then after that, immediately run this. Delete DF and delete scale data, because that will allow you to get rid of all this extra memory that you're using in your RAM to store the data frame. So on the top right corner, you should see RAM usage. It will probably be start peaking by now for you.
I did not know.
So read the data frame.
Read the data frame, drop the unnamed zero column, and then immediately run the PCA. And do you know how much RAM the Colab is giving you?
It says 86.
No, that's the disk space.
See.
Yeah, so it's giving you 12.67 GB, which might just be enough. So we will see. This is one of the reasons why single cell RNA-seq data usually runs on clusters, because as I told you, right, I have removed a lot of the data, and this is like just nothing. This is 68,000 cells. There are data sets where there are million cells. And I also removed a lot of the genes. Like if I left all those in, it will just keep crashing all the time.
Yeah.
Yeah, so you need to at least define DF and then drop that unnamed zero column because otherwise we get erroneous results. And then immediately run. And PCA will also take some time to run because it's running on like a huge data set.
Let's just rest and need to review. Nothing is done.
Okay. Yeah, if I don't know how many of you have access to like some cluster or something like that, but if you have access to clusters, I strongly recommend running these Python notebooks on that cluster. Usually, for example, if you have access to Hoffman cluster, you can just put this notebook directly on Hoffman, upload the data set that I gave you onto Hoffman, and just run it. Like you should be able to directly run. Yeah, I can believe that. It's not very easy to deal with so much data. So if it still keeps crashing, one way to get rid of all this problems is by just reducing the data frame size. Okay. So what you can do, so this is only the last minute, like the last ditch effort. So you take the data frame, which is very big, and make it a very small data frame by doing df equals df.ilog and maybe just have the first 10,000 cells or something like that. Okay, this will significantly decrease the size of your data frame and you'll be able to do PCA and the rest.
It says that initialize PCA. I don't know.
Convince Indians should not happen in India. In doing the DF. So what is this? This. Oh, no, you have to run more. So you have to drop. Oh, you drop this column. Okay. Yeah, I don't know why it's saying you haven't did it because you don't have units.
Oh, it says I cannot define the air. The air is not defined.
Huh?
It says the air is not defined. So this is just like,
Yeah, so pd.readcs will take some effort.
I see I didn't point at the license toward the
Yeah. So one quick fix you can do is do something like dot com. In what I do, you have the question? Oh, that is a process. See, that's the problem. So basically what's happening is it's crashing in between, because of which it's filling in NANs, and then then you're trying to process that. So, okay, let's do this. So all of you just forget the whole data frame with 68,000 cells and just look at the first 10,000 cells. This should reduce the size significantly and make our lives much more tractable. And when you get access to a supercomputer or a cluster, you can run the whole thing.
Where do you put the 10,000 since I missed that?
Huh?
When do you change that?
So you can just change it before. So in the beginning itself, once you've dropped the unnamed zero column, just do ef equals ef dot ilog, the first cell.
Oh,
That's it. And now you forget everything about the other 58,000 cells. Maybe even 10,000 is too much, but okay, we'll find out. Yeah, let's do 5,000. We don't have to be so ambitious. And while you're doing that and waiting for things to run, I also want to quickly show you another library that you'll probably use more if you take the single cell RNA sequencing classes, which is this thing called the UMAP. Okay. So it's another dimension reduction tool. So what it does is it takes in your whole data and then reduces it into very few dimensions. And usually people reduce it into two dimensions. So here, for example, you can very easily get it by doing pip install umap. So let me just show you how you install stuff. So you just write this pip install umap. I think, or umap learn, one of these two. Okay. So something like this, and that will allow you to install UMAP on your Colab computer and also on your local computer. And once you have that, you just have to do import UMAP. And then again, it's as simple as umap.umap. It's just a specific format where they have default values of how much neighbors you should take and so on. And the one thing I want you to focus on is the number of components. So this is where you tell the dimensionality reduction tool how many components you want in the very end. Okay, so here in this case, I'm just saying give me two components. And often you don't apply UMAP on the total actual data. So usually you apply some sort of a PCA to reduce the dimensions to a more manageable value. And then you will apply UMAP to go all the way down to 2. So here, this is the UMAP, which reduces to two components and it applies on the PCA data. And once you have that, you can plot how the UMAP looks like. But in my case, this is where my computer crashed. So I don't have the results for you. But hopefully, if you just look at the first 10,000 or so, you should be able to see some sort of a nice UMAP. Is it still crashing even if you look at the first 10,000 cells?
I'm running 5,000.
so you 5,000 is running.
Oh, okay, okay.
Hasn't crashed.
It's running. Great. So, for those of you online, it seems like people are having some success with 5,000 cells, not 10,000. So, maybe that might help you as well to stop crashing. Yeah, so PCA takes some time, okay? Because now what you're doing is you have 5,000 cells and 5,000 columns, right? And from there, you want to reduce to something like 2000 columns. That's some effort because you have to do a lot of linear algebra. So it's actually how many operations? Roughly 5,000 cube. So that's what? 125 billion operations. So if it still takes some time, then what we can also do is we can restrict the number of columns as well. We can just look at the first 100 columns or so. But hopefully it won't get to that. I think it will go through. Oh, it ran?
I think so.
Okay, that's great. Can you see what to...
Yeah.
Oh, okay, great, great. How many components is it?
The components.
You can go. Oh, 1,100. Okay, that's great. Okay, so it seems like it is running for people with 5,000 cells. So please go for df equals df dot ilog 5,000. That will actually give you some outputs.
And now I should delete this.
Yeah, so now you should immediately delete the df and the scale data because we are not going to need that for UMAP. So definitely run this code so that your RAM usage is minimal.
And now do the you.
Now and then you can do the UMAP. And for UMAP, you might want to install the UMAP Learn or UMAP. Yes. Yeah.
Yeah, friend. I asked this thing about home. I don't want to keep that.
All the cells or
I,
what is your DF?
yeah, I didn't cheat.
You didn't change it and it tracked.
Yeah.
Yeah, but it just for some reason it's running in a special way. For her, she ran all the 68,000 cells and the UMAP and everything. And it ran.
Can you make your computer lend RAM memory to the
Can you make what?
your computer can lend memory to the to add to the RAM that you have on there?
No, no, I mean, that's much harder.
Yeah.
There are ways to do it, but yeah, it's not that easy. You can't just give Google Colab some random. If your personal computer is much powerful, like for example, if you have 32 GB RAM or something, then all this will run very easily on that. Are people having success running it? You should see some very interesting pictures when you draw UMAP, especially.
I guess the UMAP was already installed.
Yeah, so if you're on Colab, many things are already installed.
Because I got the requirement for that.
Yeah, yeah. So UMAP might already be installed.
Pip is the command for
Installed.
installing.
Yes. So
So you gotta say install. It has to be pip install.
yes, so pip is a installation service. So I want to show you now why Python, one of the reasons Python is so powerful. So there is this repository called PyPy. Okay. So this is a web. So pypy.org is a collection of all the Python packages that are in existence. So for example, UMAP is one such package. And you can see there are many, many versions of UMAPs. UMAP PyTorch, apparently. I had no idea there was a PyTorch version of UMAP. UMAP Learn, UMAP Learn No TF, and so on. So there are many versions of UMAP as well. Some parametric UMAP, lensed UMAP, approx UMAP, and so on. And these are all written by different people. And to use their code, it's as simple as just doing pip install this function right here. So you can just press copy, paste it in your notebook, and now you can use all their functions that they have implemented. So it's really powerful. It's a nice central location where all the Python packages are. And they have all sorts of packages. Like there are packages to do completely like random things sometimes. So,
Right.
oh, now I'm very curious. I was not able to see the UMAP picture. So I want to see how the UMAP looks like for all of it. Oh, this is not pretty.
Yeah.
Yeah, try changing some parameters and checking. Like, usually that's the ball, that's the game. Your UMAP looks very interesting. I don't know what to make out of it. What did you get? Yeah, this looks more like it. So these are supposed to be the two clusters, and this is the third cluster or something like that. Can you all send me your UMAPs by email so I can show everyone how they look? So, and another thing to remember is PCA is a very good tool in the sense that it will really preserve the original data in a sense. You can go back to your previous data quite well. Whereas with UMAP, it modifies things quite a bit. It's a very non-linear kind of reduction process. So you cannot go back, okay, with UMAP. So UMAP changes the original data structure quite a lot. And that's how you're able to get these nice, pretty pictures.
But it doesn't change the main file. What do you mean? It doesn't change the file.
No, it doesn't change the file, but the output that you get is very, very different from the input that you gave to the UMAP.
And neighbors. I see your picture.
Okay, I want to show you some pictures that people have generated, which I could not. So this is one picture from UMAP. UMAP creates these beautiful pictures, but of course, the question is, what does it mean? In this case, I have no idea. This honestly just looks like a butterfly to me, but I don't know why it's like this. So, okay, one more UMAP. So this is how you normally expect UMAP to look like. So this is still a little bit weird, but essentially you are supposed to see three clusters. So there will be two big blobs like this, which are very close to each other. So that's one cluster. And then you'll see an offshoot here, which is supposed to be the third cluster. So if you run like a clustering algorithm on this, you're supposed to get three clusters essentially. Sorry?
It has 5,000 genes.
Yeah, this has only 5,000 genes, so that's maybe one reason. But honestly, like you probably should see three clusters even with 5,000 genes. I think we'll have to just play around with the parameters of the UMAP, like the number of neighbors and these sort of things. And if you play around with them, you can make this slightly more pretty.
It's a bit scary because then when you present data, you can present whatever you want.
Yeah, that is the beauty, right? Like you can make up new stuff. I'm only kidding, sorry. So this is one of the reasons like the integrity part is on you in the sense that with statistics and all this data analysis, right? You can tell any story you want to. Now, whether that is true or not will depend on how careful you have been with analysis. Yeah, and I think I will stop there. I hope you had fun. This was fun for me as well because I had never done anything apart from the usual sequencing pipeline. So it was fun for me to do all these random exercises, which I never actually do on the single cell RNA sequencing data. But I hope you also had fun. Like you can ask all sorts of questions because you have the biology knowledge. You can ask even more interesting questions than the ones I came up with. And with the help of Gemini and ChatGPT, you can also write code and investigate these things for yourself. I will stop there. So those of you who are taking for credit, please send me your notebooks with all the outputs.